All right, so remember that genes are sections of DNA that code for proteins. DNA is a nucleic acid, so it's made of nucleotides, and it's double stranded and a double helix. Here's a nucleotide. So we've got the sugar, the phosphate, and the base. And remember, it's the bases that make the code. So here we have a DNA molecule, so it's double stranded. And the sugars and the phosphate make the structural part or the backbone. And then between the bases, we have hydrogen bonds, and those end up holding the two sides of the DNA molecule together. Complementary base pairing, A goes to T and C goes to G. So here we have a DNA sequence, and remember that a set of three bases represents a codon. And so if I ask you to give me the complementary strand, you use complementary base pairing to give the opposite strand. So CGT ends up with GCA, and so on. DNA replication happens in S phase, and we use enzymes to unzip the DNA. So you break those hydrogen bonds, and then other enzymes come in and they create a complementary strand on both sides. So then you end up with two identical new strands of DNA. Now, finally, RNA, it's also a nucleic acid. So it's also made of nucleotides, but it's a little different. It uses a U instead of a T. It's single stranded and it's a single helix. And then, as we will see, it really is involved in helping DNA make proteins.